Good morning everyone. Today, we are discussing transduction, the process of transferring genetic material from one organism to another. We will be learning more about the observations of renowned scientists Liederberg, Tatum, and Griffith in this process. Let's get started. Bacteria reproduce through a process called binary fission. In this process, a dividing bacterial cell creates two daughter cells which are genetically identical. The interval of time between binary fission is referred to as the generation time. This can be as short as 20 minutes for some types of bacteria. Binary fission is an incredibly efficient form of reproduction for bacteria. Bacteria have three ways by which to exchange genetic material: transformation, conjugation, and transduction. Transformation is when bacteria take up free DNA from the environment. Conjugation is where DNA is passed between two bacterial cells. Transduction occurs when viruses transfer bacterial DNA from one cell to another. Although the way in which genetic material is exchanged in bacteria is varied, true sexual reproduction is absent in bacteria. Conjugation is the process in which two live bacteria interact, with the donor cell transferring DNA to the recipient cell. It was first seen in Escherichia coli in 1946 by Liederberg and Tatum. It has been used in many studies to investigate microbial genetics. Conjugation is a helpful way for bacteria to share genetic material and to adapt to their surroundings. Conjugation is the process by which bacteria exchange genetic material. For this, a special conjugation apparatus is required, referred to as the conjugation tube, pilus, or sex pilus. The donor cell containing an F plasmid produces the pilus to make contact with a recipient cell, known as the F cell, which does not possess the F plasmid. As a result of this contact, the F plasmid is transferred from the donor cell to the recipient cell. Conjugation is a form of genetic material transfer between two bacterial cells which occurs when a pilus, a thin thread-like projection from one cell, attaches itself to the recipient cell. Once the connection is made, the pilus shortens, bringing the two cells together. The F plasmid then replicates, transferring the replicated DNA to the recipient cell. Usually, the donor bacterium retains a copy of the genetic material that was shared. Conjugation is seen to be a rather conservative form of genetic transfer. Transformation is a term used to refer to bacterial genetic recombination, which was first observed in Streptococcus pneumoniae by Frederick Griffith back in 1928. In essence, transformation is the process in which bacteria cells acquire genetic material from their surrounding environment, and use this material to manifest traits not previously seen. This discovery of DNA as the genetic material, demonstrated through transformation, has been pivotal in comprehending the composition of life and its evolution. It has brought about a revolution in the scientific community, and continues to shape our understanding of biology and the future of healthcare. Transduction is a method by which bacteria can exchange genetic material. In transduction, genetic material is transported from one bacterium to another via a bacteriophage, which is a virus that particularly targets bacteria. This exchange of genetic material helps bacteria adjust and develop to their environment, making transduction a crucial part of bacterial microbiology. In 1951, Joshua Liederberg and Norton Zinder made a major breakthrough in their research by discovering genetic recombination of Salmonella typhimurium, something that had remained undetected until their investigation. This discovery provided insight into the development of antibiotic resistance in bacteria and paved the way for more effective treatments to combat bacterial infections. This slide is focused on MCQs. We can identify 4 possible choices for this particular question: conjugation, transformation, transduction, and binary fission. Which answer do you think is correct? Bacteria have various sizes and shapes, yet share some common features. One of these is the pilus, a hair-like strand extending from the cell's surface which allows the bacteria to bind to other cells. This strand is also utilized in transformation, binary fission, transduction and conjugation, all of which are necessary for bacteria reproduction and dissemination. Bacteriophages are viruses that can facilitate the transfer of genetic material to bacteria, making them capable of rapidly adjusting to their environment. 4 steps are involved in this process: conjugation, transduction, transformation, and binary fission. Conjugation refers to the exchange of genetic material between 2 bacteria. Transduction happens when a bacteriophage transfers genetic material from one bacteria to another. Transformation consists of bacteria taking up genetic material from their environment. Finally, binary fission is the simplest form of reproduction, wherein one cell splits into 2 replicated daughter cells. Bacteria are a vital aspect of our lives. They provide us with a plethora of benefits. They allow us to create vital vitamins and proteins, aid in digestion, and help regulate our body's balance. Similarly, they form a substantial portion of our respiratory systems, contributing to oxygen circulation. Additionally, bacteria in the soil help to decompose organic matter and feed plants with important nutrients. Without bacteria, the world would be a much different place. Bacteria hold a crucial place in our lives, but their importance often goes unrecognized. They are part of the environment, and thus affected by its alterations. Bacteria found in soil, for instance, are needed for the soil's fertility and are essential to the decomposition of organic matter. Inside our bodies, bacteria assist with digestion and our protection against sickness. In conclusion, these microorganisms provide us with huge advantages, and they definitely deserve their title of friends. Knowing the bacteria responsible for certain diseases is invaluable to protecting public health and preventing potential epidemics. Clostridium tetani causes tetanus, Clostridium botulinum causes botulism, Vibrio cholera causes cholera, Salmonella typhi causes typhoid, Corynebacterium diphtheriae causes diphtheria, Mycobacterium tuberculosis causes tuberculosis, Diplococcus pneumonia causes pneumonia, Mycobacterium leprae causes leprosy, Neisseria gonorrhea causes gonorrhea and Treponema pallidum causes syphilis. Tetanus is an infection caused by the bacterium Clostridium tetani. It is usually acquired through contact with the spores of the bacteria which occur in soil and animal feces. Symptoms of tetanus include muscle spasms, difficulty swallowing, and even death in serious cases. It is important to be aware of the potential risks of tetanus and to get medical attention if you think you may have been affected. Vaccines exist to help shield against tetanus. Botulism, commonly known as Clostridium botulinum, is a serious and potentially fatal illness that is usually contracted by ingestion of contaminated food. It is caused by a toxin produced by the bacteria which affects the nervous system, potentially leading to paralysis or death. Early diagnosis and treatment of botulism is essential in avoiding serious health risks. Vibrio cholerae is the bacterium responsible for causing the disease cholera. Cholera is a severe, infectious disease which is contracted by consuming water or food contaminated with the bacteria. If left untreated, it can result in serious health complications and even death. Proper treatment requires replenishing the body's fluids and administering a course of antibiotics. Salmonella typhi, more commonly known as typhoid, is an infection caused by the bacterium of the same name. It can cause fever, abdominal pain, and sometimes diarrhea in humans. It is spread through contaminated food and water, and treatment includes antibiotics. It is essential to be aware of this infection and take all necessary precautions to avoid contracting it. Corynebacterium diphtheriae, commonly known as diphtheria, is a potentially fatal bacterial infection spread mainly via person-to-person contact. It is caused by the C. diphtheriae bacterium, with symptoms including sore throat, difficulty breathing, and a thick gray membrane covering the throat or nose. Fortunately, it is treatable with antibiotics, and prevention is possible through immunization. Early detection is essential for successful treatment and preventing the spread of the infection. We will discuss the symptoms of diphtheria, a serious bacterial infection. Characteristic of the illness is a thick, gray coating forming in the back of the throat, making it difficult to swallow or breathe. Fever, chills, weakness, and swollen glands in the neck can occur. If left untreated, the infection can potentially be life-threatening. Early symptoms to be aware of are a sore throat, low-grade fever, and tiredness. To prevent its further spread, medical treatment must be sought if any of these symptoms appear. Concerning Mycobacterium tuberculosis, commonly referred to as TB, it is an infection caused by a type of bacteria. If left untreated, it could be fatal. This bacteria is transmissible between persons, with the symptoms ranging from fever, chest pain to coughing up blood. Therefore, it is imperative to identify and treat TB as soon as possible to avoid passing it to others. Diplococcus pneumonia is a type of bacteria that can cause an infection of the lung known as pneumonia. It typically resides in the nose, throat, and mouth, making it a frequent cause of respiratory illnesses in adults. This bacteria can be transmitted through droplets when a person coughs or sneezes, making it contagious. Treatment of pneumonia resulting from this bacteria can be done swiftly with antibiotics. However, if left untreated, it could potentially lead to serious medical complications or even death. Leprosy, also known as Mycobacterium leprae, is a highly contagious and devastating infectious disease that affects millions of people across the globe. It is a chronic condition caused by the Mycobacterium leprae bacterium that weakens the body's mucous membranes and skin, causing lesions that could lead to disfigurement and paralysis. Although it can be cured, especially with early diagnosis and treatment, it remains a challenge in several parts of the world due to the scarcity of resources and lack of awareness. Neisseria gonorrhoeae, commonly known as gonorrhea, is caused by the bacterium Neisseria gonorrhoeae. It is one of the most common bacterial infections in humans and can have serious consequences if not treated. Symptoms may include abnormal discharge from the genital area, painful urination, and in women, pelvic inflammatory disease. If not treated, gonorrhea can cause infertility, ectopic pregnancy, and even death. It is recommended to seek medical help if infection is suspected, as treatment is available. Bacteria are single-celled microscopic organisms that can be found in almost any environment. Of special interest is Treponema pallidum, which is responsible for the sexually transmitted infection known as syphilis. Syphilis can be treated with medical interventions, but it is important to be aware of the symptoms and to always practice safe sex in order to avoid contraction of the disease. Bacteria can be serious offenders when it comes to causing diseases in plants. Blight of rice is caused by Xanthomonas oryzae. Citrus canker is brought on by X. axonopodes pv. Citri, and crown gall of apples and pears is caused by Agrobacterium tumefaciens. Plant diseases caused by bacteria can have a major impact on crop yields, making them both damaging and expensive for those in the agricultural industry. Examining bacteria, it is apparent that its effects on plants, especially rice, are substantial. Blight of rice is a prominent problem, impacting thousands of hectares of cultivated rice fields. Thus, it is necessary to understand the effects of the blight and take steps to prevent or reduce its influence. Citrus canker is a disease caused by a bacterial infection that typically affects citrus fruits, like oranges, lemons, limes, and grapefruits. Symptoms include discoloration and pitting of the peel, defoliation of leaves, premature fruit drop, economic loss, and potential death of the tree. Therefore, it is important to recognize the signs of citrus canker and take the necessary measures to limit its spread. Crown gall of apple plants is a disease caused by the bacterium Agrobacterium tumefaciens. It enters the cells of the apple tree, multiplying and forming galls or tumors on the tree's surface. These galls affect the structure of the root system, reducing the tree's productivity and quality. To avoid crown gall, gardeners should purchase apple trees from a reliable source and maintain soil free from bacteria. Bacteria is responsible for causing crown gall, a disfiguring and unattractive disease. These bacteria enter the pear tree through any available wounds such as the ones caused by pruning. Once inside the tree, the bacteria starts to grow and divide, which results in the formation of galls around the infected wound. These galls gradually increase in size and distort the stem of the tree, restricting the flow of water and essential nutrients, leading to wilting and leaf drop. Therefore, prevention and treatment are both necessary for the management of crown gall. Pruning wounds should be covered and sealed with protective wax to prevent contamination. Whenever possible, choose pear varieties that are resistant to the disease in order to reduce the risk of infection. Chemical control is also available and it requires regular spraying applications. Bacteria can indeed be our friends, even in our own backyards. Moving forward, let's explore how this community center managed to bring together members from various parts of town to promote this worthy cause. Bioscience has become a major component in the operation of modern laboratories. Advanced technology has allowed activities such as biomining, biosensors, medical diagnostics, food operations, biotechnology, genetic engineering and fermentation to be conducted with greater efficiency and accuracy. Bacteria are capable of unlocking the potential of these operations and creating a more cost-effective process. Through the use of bacteria, compounds can be extracted from natural sources, contaminants can be detected in food and water, and medical screenings can be done. All of these activities lead to greater quality control and safety for end users, and they also create new opportunities in the field of biotechnology. Microbes have a wide range of applications in the mining industry, particularly through biomining. This process can significantly lower production costs by more than half. Additionally, biosensors can detect biologically active toxic pollutants by analyzing the DNA components of bacteria, presenting new ways to monitor and reduce toxins in the environment. Answer: Clostridium botulinum is the answer to this multi-choice question. Found in soil, this single-celled organism can cause a severe form of food poisoning. Botulism: a serious, potentially fatal illness. It is therefore important to take precautionary measures in order to avoid contact and contamination. Crown gall disease is a plant disease caused by bacteria from several genera, including Agrobacterium tumefaciens, Xanthomonas, Diplococcus, and Mycobacterium. Symptoms of the disease are tumors and growths on stem tissue and roots, resulting in stunted growth, wilting, reduced yields, and even death. To prevent and control the disease, a combination of cultural, chemical, and biological control methods must be employed. Mycobacterium tuberculosis is a species of pathogenic bacteria which can cause a range of diseases, including tuberculosis and leprosy, as well as other lesser-known diseases. It is one of the most common causes of death in the world. However, medical treatments are available for those suffering from diseases caused by this bacteria. Bacteria can cause a range of diseases, and one of them is citrus canker disease, which affects apple, lemon, pear, and tomato crops, all crops important to our economy and diets. We must be mindful of citrus canker and the damage it can cause, in order to be proactive in preventing its transmission and protecting our crops. Thank you for your time and attention.